Okay, so let's look now at some of the principles. What are the principles on which simulation neuroscience rests? So the first important principle is that we have to do dense reconstructions, dense, very detailed reconstructions, and we have to do it from sparse data. Okay, so I just gave you an idea, a glimpse at how much data there really is to measure. And if anybody has to do a calculation, you'll realize that it will take the entire human race thousands of years if you wanted to map out all of those things in all of those different conditions. So we will forever have sparse data. No matter how much data we collect, it will forever be sparse in terms of experimental. So the problem we have to solve is, how do we get a complete picture from sparse data? So the first principle of simulation neuroscience is we have to establish this strategy from going to a little bit of data to using whatever knowledge we have of how the pieces fit together to build algorithms and algorithmically build the circuit. Now, this first principle is not a new principle. It's extremely old. It's the basis, actually, of our entire society today of information and communication technologies. Let me try and explain this in another way. Imagine that you have a TV screen, okay, and the channel is unplugged. So it's a white noise. So basically every pixel is independent of every pixel. Okay? And now I want to transmit this image to you. I want to tell you what is this image. I need to give you a complete picture of what this image is. Based on Claude Shannon's work from the 40s, we know the theory here. It's very clear. I have to measure. If this is a random system, if this is a random image or a random system, I have to measure every single pixel. I have no way out but to measure every pixel if I want to tell you about this complete story here. Okay? But it's only if I have a random system. If I now have an organized system where there's a very clear structure, the theory shows you absolutely clearly that I don't need to transmit every pixel to here. I don't need to transmit every single pixel. So I don't have to tell you what every pixel is here, because there's relationships between all these pixels, and that's the basis of image compression. So this is order. So in this case, if I have a random system, I don't need any intelligence. I just have to copy these pixels across. So I don't need an algorithm here. Okay? I don't need an algorithm. I just need to transfer the data. But if I have the order in the system, I need a rule how to rebuild it. If I'm only going to send a few of these pixels across, and I want to rebuild this picture, I need an algorithm. Okay? I need some rule that will rebuild the relationships that are in this picture. Okay, so in a way, with a random system, I need to measure everything, and I do it blindly. I don't understand anything. I just have to copy it from here to here. In an ordered system, I have to understand the order, and I have to package it into an algorithm, and then I can send it to you, and I can reconstruct it. Basically, the important message is that dense reconstructing from sparse data is not just because we have to, because we'll forever have sparse data. It is because it's science. It's actually not scientific to say that we need to go and measure everything in the brain. Okay? Only if it's a random system do we need to measure every pixel. You don't need any principles. You don't have to understand very much. And you can close your eyes and blindly map one thing to the next. So when there is an objection that says, oh, you can't reconstruct or simulate a cell because we don't know everything about the cell, that's not a scientific statement. The challenge is to find the minimum data you need. The smallest data set, not the largest data set. The real science is to say, what's the least amount of data I need to be able to reconstruct it? That's a compression algorithm. It's the inverse. What's the least amount of data that I need to transmit to you so you can rebuild the image? Okay. In that case, you do not need to measure everything. You need principles, and that's what drives simulation neuroscience. And then you algorithmically reconstruct it. So simulation neuroscience is fundamentally about finding those principles, packaging them into algorithms, and then reconstructing. That's the first principle of simulation neuroscience. The second principle is actually also quite similar to computer and software engineering. It's a bottom-up reconstruction process. And it actually has very strict rules. And the rules are, first of all, you've got to follow the biological principles. You don't try and invent how you think the brain should look or the neuron should look. You measure and you use as much of the data that is available, and where it's not available, you have a hypothesis of what is available. And you can then test that hypothesis. You build the smallest components first. So you really start atomic, if you wish. You start at the ion channel to build a neuron. You don't start at the neuron and try and make a neuron behave and then say, oh, let me go back and build ion channels. You start at the very bottom and you build upwards. Okay? And a key thing, which is the same as what you do in computer and software engineering, you build modules and cells and functions, and you freeze your variables, and you freeze them, and then you combine them. If you go to Python, it's so beautiful. It's combinatorial, because I just build these and then I can consume them later. But in order to do that, I freeze the components, and then I can combine the components. And the other important thing, and very different from computational neuroscience, for example, is you validate upwards. In other words, you validate what emerges when you combine things. You don't try to fit your data. You never fit the data on looking up at an emergent property. So, for example, you wouldn't try to say, look, my brain is not speaking, so I'm going to adjust my ion channels until they speak. You'll be adjusting ion channels forever. Okay, and that's it's an intractable problem. So you've got to come down to the emergent property as close to that parameter as possible. For an example, an ion channel, you want to build an ion channel, you look at the behavior of an ion channel. You don't look at the behavior of a neuron to build an ion channel. So you always do it at the bottom, you freeze it, and then when you say, okay, I've got my ion channels, I want to know what they do, I put them in a neuron, then I look at the neuronal behavior, and that way I can see how they play out when they are being used. If you find that the neuron, you can never replicate the neuron's behavior, then you can say, maybe I got my ion channels wrong. Maybe I don't have the right ion channels, and so on. So that's where we begin the cycle, which we're going to learn a lot about in the course of this and other lectures. The guiding principle here is that if you build it right, it will automatically behave right. Okay, so you build bottom up, you build the components, you freeze them. If this is valid in terms of the behavior, even if you don't know all the pieces, but if a neuron behaves right and you put it in a circuit, you can talk about what neurons do. If the circuit behaves right and you put it into a brain, you can talk about what the circuit does in that brain. Okay, so you don't try and tweak the model to behave right. If the neuron, if you built it right, it automatically behaves right. The third, there's of course many, but the third principle is that it's iterative reconstruction and testing. So you get your experimental data, you get your literature, you go and find all the papers you can, you mine all the facts you possibly can, and you try and build a unifying model of a neuron. You find the ion channels, you find the morphologies, the structures, the physiology, any data that you have about that neuron you collect. And then the challenge is, how do you package that into a simplest algorithm that will then reconstruct it, that is consistent with all the data that you gathered? You obtain your model, and then you interrogate it anatomically. You look at it and you see, okay, I got it right. It looks like a real neuron. I do the statistics between my reconstructed neuron and the real neuron, and I see that they match. I look at where the ion channels are, they match, and so on. And then you simulate it to see if it behaves the way that it does in the real tissue. And then you do an analysis, where you do the systematic comparison, and you get your errors. It's not going to work the first time. In fact, the goal is to do it very fast, fail very fast, identify where you may have got the wrong data or the wrong assumptions, and then you build it again. And you keep going through the cycle as many times as you possibly can. In fact, it's an endless loop. You keep going and integrating and perfecting it. But with each cycle, this model replicates the tissue more and more accurately. If you follow this process, you can't go left, you can't go right, you can't go backwards, you can only go forwards. So the model can only improve each and every time that you integrate it. So to illustrate this, let's just imagine that you step inside a forest of neurons. And you look up at the neurons, there's thousands of neurons, millions of synapses. And you look around and you say, I wonder how these neurons connect. I mean, how are they connecting with each other? Who is connected with each other? Where are all these? So how do you approach this problem? Of course, you can try. Let's take an electron microscope and go and measure everything. That's, of course, one approach, but if you actually look at it, you will be measuring for a very, very long time. But there is another approach, and the way that we do it in simulation neuroscience, that we say, okay, let's just look at what we know. Let's start off, and let's imagine that we have a whole series of cells, neurons. Okay. And of course, there are different kinds of neurons. You have many different kinds of neurons, and each one looks different, behaves different, and it can, of course, connect differently to the others. So how do you approach this problem? Well, you can say, what do we know? And maybe there's only one experiment that tells you how this one cell, this one type of cell, connects with the others. There are potentially thousands of different types of connections here, but you only have one experimental data point. And this experimental data point, you go and you see that the axon that goes to connect all these neurons does this. And you look at this and you analyze the data here and you say, oh, I can see a principle. First principle, the axon somehow touches every neuron. Okay? So before I look at who's connected, I just see that the way that this axon goes, it's like what we call a tabula rasa. It goes and it touches every single neuron. And then I look at this one neuron and I say, oh, I see that when it touches onto another cell, it actually puts lots of little synapses on it. It never puts just one synapse. Okay? It never puts just one synapse. It puts lots of them. So I see principle number 2. It's a multi-synapse rule. Okay? And I think, oh, that's interesting, but what else can I learn by looking at this one experiment? I see principle number 3. I see, wow, if I look at the axon of this cell, and I look at it very carefully, I see that it's got places called boutons, which is where it can form synapses. So I think, how many are there? And what is the space between them? And then I see, okay, that will limit how many neurons this one can contact, right? Because if I have an axon that only has 2, then it's of course going to have a very different connectivity than I have an axon that has many boutons. So now I have a bouton principle. It's a physical limitation. If I have lots of boutons, if I have a neuron with an axon with lots of boutons, then I can contact lots of cells. If I have one, I can contact one cell. So these are 3 principles. Now, there are more principles, but let's just assume you say, okay, I like these 3 principles. I'm going to see if I can rebuild the tissue just with these 3 principles. So we package them into an algorithm. Okay. And digitally, we reconstruct this tissue. Now you have 2 possible scenarios. It succeeds or it fails. Succeeds means that you have a look at it and then somebody tells you, hey, you know what? There are 10 other experiments out there that looked at how this cell connects and this cell connects. And you have other data that you can check it with. And then when you look at that, you see that your reconstructed tissue now reproduces all those other experiments. So you think, wow, I mean, I looked at one cell, I applied 3 principles of how they should connect with each other, and they reproduced what other people had done in other laboratories all over the world. That's a success. But what's important to understand in this success is that you haven't really gained any new knowledge, but you have gained a new understanding. You understand how your principles interact. You understand that these principles for this neuron applies to all of them. 2 very concrete new understandings. It's not new knowledge because this knowledge existed. People had measured this, we saw this, it was just fragmented knowledge, but you never had that integrated, unified view. So when it succeeds, you understand better. I'm an experimental neuroscientist, and I've looked under a microscope for 20 years. And when I put this together, in a single reconstruction, I understand what I was doing for all those 20 years. Because you can see this as an integrated answer or perspective on what the pieces are that you've been collecting for so long. Now the other scenario is it fails. I do this, I check it again, somebody comes into the door and they say, look, I have an experiment and it shows me that this cell, it really doesn't follow those principles and it connects in a completely different way. And then I have new knowledge. I know that this principles are not enough. There have to be new principles. There have to be some other rules that will apply, whether there are exceptions or whether that these principles have to be modified, but there has to be something else that has to be understood. And then one can start investigating. And that triggers, when you fail, it triggers new experiments. Because you have to test now, how would an algorithm reconcile these differences? Another way to describe this is, imagine that this algorithm works in one part of the brain, and it explains all the way the neurons connect. Now I go to another part of the brain and it doesn't work there. And you think, oh, that's terrible. No, that's great, because it tells you that in that other part of the brain, there's something else going on. And that's something else nobody knew about before, because you had too much fragmented knowledge. And that is when failing really leads to an advancement of knowledge, new knowledge, because it triggers a completely new perspective. It actually shows you the boundaries of our current knowledge. You get to the end, you say, I've taken all the data, all the principles, and I cannot explain how this happens. Then you're at the frontier. And at that frontier, you can either do theory, you can do experiment, you can iteratively test, but there are many ways to advance it. And this is a very exciting approach to advancing knowledge. Okay, so we looked at these three principles of on which simulation neuroscience is based.